So welcome everyone to the NextGen Precision Health Discovery Series. This is our flagship seminar series, and we have a speaker today that it's a big honor to introduce. He's actually my first science mentor as I was transitioning from being predominantly a clinician to trying to learn how to be a physician scientist. So Dr. Arthur Burghes is a professor in the Department of Biological Chemistry and Pharmacology in the College of Medicine at Ohio State. He did his Ph.D. training at the University of London and then subsequently did a fellowship, postdoc fellowship at the Hospital for Sick Children in Toronto, and then joined the faculty at Ohio State and has been there since. In 2019 was awarded Distinguished Professor at OSU, and his work has largely been focused on understanding spinal muscular atrophy, but he actually started in Duchenne muscular dystrophy, and that's what brought him to OSU as faculty. So before I forget, I do want to mention that we are going to try something new today. So we have the Mentimeter platform to be able to add questions throughout the lecture. So the info is on the yellow board here. And then also, I think do we have a, so you can use this QR code to also get to the website and enter your questions. And so please enter those as we go along. We'll also, you can put those in the chat if you're on Zoom, and also in person, we'll also have the mic. But give this a shot. Let's see how that works. And so I won't delay any longer. I'll hand off the mic to Dr. Burgess, and let's welcome him.
Well, it's great to be here, and I will try and stay between the lines, but I have a tendency of wandering around. Now the mic is on. So start one. Okay, so I'm going to start here. So my title is obviously a little facetious. So King Arthur's Knights is everybody that's ever been in my lab, plus any associates that we've worked with. And the search for the Holy Grail is to understand and get therapies for SMA and the mechanism of SMA. So obviously that's filched off Monty Python. And for the younger audience, Monty Python is a British TV comedy from years ago, and I would recommend watching it highly. My conflicts, I consult for Novartis Gene Therapy and Mammoth Biosciences. So SMA is what I want to talk to you today about. It's an autosomal recessive motor neuron disease, at least predominantly motor neuron. It has an incidence of 1 in 10,000 live births in the Caucasian population. The frequency is not the same in all populations, has a lower frequency in the African American population, for instance. It results in loss of the alpha motor neurons in the anterior horn of the spinal cord. When you lose your motor neurons, you get atrophy of the limb and trunk muscles, and it's called proximal spinal muscular atrophy for the simple reason that it affects proximal muscles more than distal muscles. It can be divided into, well, I've only got 4 here. There's also a type 0, but 4 different types that basically have different severities when untreated. So type 1, babies never sit, they can't walk, and they can't even lift their head very well. And they die before the age of 2 if untreated. Type 2 is slightly milder, type 3 even more mild, and type 4 has an onset in adults and is very mild. The one characteristic feature that you can find clinically is basically fibrillations, or basically spontaneous firing of the muscle, in particular for type 1 cases. This is also accompanied by a decrease in the CMAP, or compound muscle action potential, and also the number of motor units going to a muscle. In type 3, that is less pronounced in that the motor neuron can sprout and reinnervate, which gives an enlarged motor unit as well. This is all in the human. The SMA region was originally described as an inverted repeat, and yes, I go way back to doing the linkage studies in this disorder, which I'm not going to bore you with. But basically, the inverted repeat actually is probably not an inverted repeat in a large number of people, and below is a map that we assembled using a series of back clones, and they all derived from a single individual, and insisting that they had large 35 kb overlaps and that all the polymorphic variants matched so that you could determine that it was from the same chromosome. And if you look at this, you can simply see, I've not got my laser on. You can see that the SMN2 and SMN1 gene is actually in the same orientation. So it's not an inverted repeat here. It is actually a straight tandem repeat with an insertion in the middle that doesn't contain any SMN genes. Is this the structure in everybody? Probably not. This region is still a bit of a mess in the human genome, even though we've apparently completed sequencing everything and we know everything. We don't. There's still some mistakes in this region. How is SMA caused? Well, there's two SMN genes. They both produce some protein, but there is a critical difference between the SMN1 and SMN2 genes, and that is this C to T change. And in SMA, you lose the SMN1 gene, so you're reliant on SMN2. And just before I go on, a person critical to identifying this thing is listed here, this Lawson CL guy that you may know. And he's also first author on the other paper that describes this. This actually confirmed when he was a postdoc that this was the critical difference between the SMN1 and SMN2 gene. So when SMN1 goes or is mutated, then you're reliant on SMN2. And that partially compensates for the loss of SMN1. If you lose both SMN1 and SMN2, it's a complete lethal phenotype. And you can tell that in humans because that chromosome does exist, but it's never found as a homozygous deletion of both genes. So the reason SMA presents is because SMN2 is present in the human population and produces a little bit of SMN, and this mutation here, the C to T change, causes the exon 7 to be inefficiently incorporated, which leads to the truncated protein, which is rapidly degraded, and then this little bit of protein gets through and can compensate. The more copies of SMN2, the milder the phenotype. And that's shown here. This is from actually the Spanish group, Tizano. And you can see that most of the type 1 patients have 2 copies of SMN2. If you go to the milder phenotype, type 2 have 3 copies of SMN2, and type 3 is a mixed bag. This shows the whole world population in every study. But one question that comes up is, what is the reason for these exceptions? For instance, over here there are some 3-copy cases, and over here there are mild cases with 2 copies of SMN2. Well, the first question that comes up is, are all the SMN2s identical? And the simple answer to that is no. So if you have this SMN-C859 G to C change, it's a positive modifier of the splicing of SMN2, resulting in more full-length SMN and therefore more SMN protein. And if you actually go to this again is in the Spanish population, you can see there are no type 1s that ever have this mutation, regardless of their SMN2 copy number. If you go to the type 2s, you can see this is in the heterozygous state, and 4 out of 24 have them. If you go into the type 3s, it's actually in a homozygous state. Now, this is not the only modifier in SMA. There are other modifiers that occur, and some of these are in the SMN2 gene, but a lot of them do lie outside of the SMN gene, and we still really don't know which ones they are. Yes, I know about plastins being reported as a modifier. I'm just not one of the people that agree with that. One of the first things you want to do when trying to go on a translational trip is to make some model that you can use. So the first thing that occurred is the mouse gene, and actually all other species, do not contain an SMN2 gene. So man is the only beast with an SMN2 and an SMN1 gene. Chimpanzees and the monkeys do have 2 SMN1 genes, but they don't have an SMN2 gene. Mice and all other organisms have a single copy of SMN. So when you delete SMN, which was a great rush to do after cloning the gene, you actually make an embryonic lethal. We did not do that, and we didn't do it deliberately because we already knew from the human data that you could never get a homozygous deletion of both SMN1 and SMN2. So we decided we took the PAC 215P15, which contained the whole SMN2 gene, and it's conveniently on basically a 35.5 kb BAM H1 fragment. So we cut the PAC with BAM plus another enzyme to make these fragments smaller, and isolated from a pulse field gel, which if anyone really wants to know, I will explain later, the 35.5 kb band, and injected into mice. And from that, we got founders, and the first founder is 89, which had a single copy of SMN2, and the second founder was 566, which is got 8 copies of SMN2 on a single chromosome. We then contacted Michael Sednetter in Germany, who'd made the knockout and published it in PNAS. And being of Germanic origin, he insisted on using Lufthansa to fly the mice to the US. And so we took a road trip from Columbus to Chicago. Unfortunately, my car is a BMW at the time, so we could accommodate the mice in the way they were accustomed to and drive them to Columbus in comfort. They arrived in Columbus. They weren't actually the youngest mice, so we had to actually force the breeding a little bit. And so what we did is took the males and crossed them right away to the females that we had in the lab that were the FEBs with the SMN2 in them. And FEBs actually breed much nicer than C57 blacks. And so that got us the mice with the deletion. So that actually, this just summarizes the mouse models. So loss of SMN entirely is embryonic lethal. 1 copy of SMN2 on an SMN minus background does not rescue. It's lethal again. Then we come to the 2 copies of SMN2, so the line 89 homozygous with the minus background. So the first SMA mouse in Columbus was born, of course, just around Christmas time, just for convenience, so that you have to go in Christmas. And it had a very severe phenotype, dying at 5.16 days when it had 2 copies of SMN2. Of course, the 0.16 is vital in that statistic. not really. Then we had mice with 8 or 16 copies of SMN2, depending on which was from the other line, the 566 line. These mice had a completely normal lifespan. The only phenotype they showed was that their tail was slightly shorter. How that translates into man, I have zero idea. So those were the first SMA mice, and the first important thing that we did do in the first paper was show that there was motor neuron loss. And this becomes important later on. Hish Lee in Taiwan produced mice that also had 2 copies of SMN2, and they had a severe phenotype. He made the deletion such that exon 7 was knocked out on it. They lived actually for about 10 days. The only problem with those mice is they actually don't have loss of motor neurons. And that's slightly an issue in that the classic phenotype of SMA in humans is loss of motor neurons. So we took these mice and we actually bred the SMN lacking exon 7 as a transgene onto these mice. And the reason we did this at the time was not to make the best model of SMA, but actually to show that the delta 7 was not a toxic product, even if it produced a little bit of protein, which must be exceptionally small, but it wasn't a toxic. So when we put it onto the mice background, these mice jumped from 5.16 days to 14 days. So that was the basis, and those are the mice that have been used in a large number of studies developing therapeutics and form the basic. And the difference between dying at 5.16 and 14 days is quite significant when you're trying to actually test therapies. We did also produce a milder mice model, which is a mild SMN allele, and subsequently Rashmi Kathori's group produced the 2B minus mouse, where there's a disruption in the exon 7 of the mouse gene and across from a deletion. And this actually has a relatively severe phenotype as well. It lives slightly longer than the delta 7 mice. Again, this person called Dave Arnold came into the lab. You may know him, I don't know. And we wanted to actually take the electrophysiology that was used in the clinic and adapt it to the mice. So lo and behold, SMA mice have fibrillations, as shown here from Dave's slide. The CMAP is reduced in SMA mice, and also the MUNI number, or the motor unit number, is also reduced. So this gave us a measure where we could do it on live mice and not kill them every time we were doing the electrophysiology. Okay, now I'm going to go right into therapy. And I'm going to say some things, and I'm going to go over them a bit quickly because a lot of this is now known. So SMA is caused by too little SMN. So what do? You get more SMN back in there. So very simplistic. We're not very good at thinking in complicated manner, so we keep it simple. And all 3 of the therapies that have been developed all correct the mouse model, but result in mice that either lack or have short tails. And so we've created lots of hamsters in all these treatments. I don't, again, know how this relates to humans. So what are the treatments that are developed? So first is the drug compound, which was developed originally from a screen by PTC and then bought by Roche. One interesting thing about this chemical is And one of the differences in the screens of the chemical, it's not that anybody hadn't thought of this before. The mini-gene was first developed by Elliot Androphy's lab, which of course Chris Lawson was a member of. And then it got adapted and it got used in some of these assays. The critical thing is they took bad compounds and did a lot of chemistry on them to make them better. In other words, the libraries don't really possess modifiers of splicing. That's one of the keys there. And they had a major impact on SMA mice. So the mice went from 14 to 150 days plus. For some reason, because it's a company, you can't work past 150 days, so they just stop giving them the drug and they die. So went crazy, we come on in every weekend dosing mice. And this was reported in Science by Narshkin et al. And eventually, with more chemical modifications, has resulted in RG7916 or risdiplam. You can work out the actual commercial name of it. The second one I want to talk about is the antisense oligos. And these are antisense that actually bind and block a negative regulator of splicing. It's called ISSN1. And it blocks those, allowing the incorporation of exon 7. The papers from Adrian's Kraner's group and Ionis used an MOE chemistry. The ones that we used were actually a morpholino chemistry. So the end result was the development of nusinersen, which was approved in 2016. And that is one of the treatments that's now in place. Then lastly, and I'll probably concentrate a bit more on this, is the gene therapy that was developed basically between my lab and Brian Casper's lab at the Nationwide Children's, with Brian having the AAV expertise, not me. So AAV9 was discovered to cross the blood-brain barrier. So we made AAV9 SMN. And the first thing I will mention, which is not actually in a lot of the papers, is that we did not put any tags or anything on it. And that was my influence, because we never did it in the transgenes, and I was nervous of disrupting the function of the protein. It so happens that the group of Martine Barkatz also made an AAV9 SMN construct, but they actually put a tag on the C terminus of the SMN. And I got a phone call one day in the lab. I answered the phone, and it was the technician from France that worked for Martine and said, "I can't get this to work, it never works." And I said, "Have you tagged the SMN?" And he said, "Yes." So as soon as they took the tag off, it worked. So be careful putting tags on proteins. They're very convenient, but they don't always work. They can disrupt the function. So a long story short, the papers got published showing that it did rescue mice, and then it was improved in 2019 for delivery via the bloodstream. It also actually does work via intrathecal injections, but that's still under, still in clinical trial in the phase 3. So this shows the actual compound that PTC developed, and this is one of the early ones, so it's called C3. And the first one simply shows the actually incorporation of exon 7. So this is actually using a kind of fancy assay, and you can see that the amount of full length goes up as the compound increases in concentration. And that's shown on the gel below. You can see right away that the full length SMN here is increased. And if you actually use a protein assay, again, it's increased. And if you sit down at the microscope and count these things called gems, which are nuclear deposits of SMN, that also increases. The survival of the mice, that goes up when you dose these. Again, they stop at this point. It's too long, so they want their weekends back. And this shows the weight. For some reason, when you do these corrections, the weight of the mice is always slightly less. If you go to the actual, this is actually with the morpholinos, because this is the way we did it. So you can inject into this. This shows the different concentration of antisense morpholino, 27, 81 grams, and 81 grams. This shows spinal cord. The top band is full length. The bottom band is delta 7. So this is the scrambled oligo, and you can see it's hardly producing any full-length SMN. And then when you give the antisense, you have an increase, even out to 65 days. And at 81 grams, it's even more marked, and it also occurs in brain. If you look at the protein, as you'd expect, it's increased and drops with time as the ASO dilutes out, because this is a single injection into the ventricle of the mouse, starting at P0. If you look at the survival, this shows the survival curves for the different concentrations of ASO used, and this shows the actual time the median time that the animals lived. So you can get mice living 100 days with a single injection of the ASO. Obviously, you would inject in a person multiple times. You would not put it into the ventricle, you put it in intrathecally. It's just we're trying to do the equivalent in the mouse. Yes, there are two chemistries. This is the MOE background, which is actually in the clinic. And this is the PMO background that is not in the clinic, but what did show effectiveness. And the one thing I will say is that the morpholino in our hands has much less toxicity, so you can go to much higher concentrations of it than the MOE. If you go to patients, what does it look like? So there are two groups of patients here. So this line, the red line, is actually patients that have symptoms and are treated with the ASO. This, in unreadable print, is the days afterwards. I apologize for the unreadable print. I should get this slide and redo it. This is the pre-symptomatic case with 3 copies of SMN2 or 2 copies of SMN2. And this compares those two groups. The first thing you notice is that very early treatment, pre-symptomatically, is much more effective than treatment symptomatically. And lo and behold, this is a kid from one of the trials on the train from New York, and the reference is given there. So the treatment is started at 15 days. It's 15 days after birth. And you can see that this, at 14 months old, this girl is walking with assistance. But remember, an SMA child never sits. So this is a remarkable impact. This is the pre-symptomatic treatment. So pre-symptomatic should be like, it's not really pre-symptomatic. I mean, be careful with that term. It's used because the physician can't see anything. If you actually measure neurofilament, there has been damage at this point. So depending on how you define pre-symptomatic. If we go to gene therapy, this shows the self-complementary AAV vector with the chicken actin promoter driving expression. It has an SV40 intron, and that SV40 intron has no enhancer in it. The actual endogenous chicken beta actor intron does have an enhancer as it, and that is in some vectors. This particular vector didn't. Again, it's going to be tested in the delta 7 mouse. And the first thing I'm going to point to is the survival curve, which is here. I know this is not the survival curve in the actual paper. This is the revised survival curve when all the data was taken if you take all the days that died after the paper was accepted and all the ones that died before that. The curve shown in the actual paper has caused a lot of stir. It resulted in the retraction of the paper by the editors. Do I agree with that? No, I don't agree with that. This is the actual data complete, as in the mice still survive for 197 days. That's a long time to survive and a pretty significant effect. That does not change the quality of the data, in my opinion. Should we have been more careful and actually made sure that we revised the paper after they'd asked for primate studies and everything else? Yes, I completely accept that, and so does Brian. This data was not collected in my lab. It was collected in Brian Casper's lab when he's there, but I see no reason to actually pull the paper because of it. The other thing about this paper is that we actually work with Mark Rich, and you can do electrophysiology where you look at the actual end plate potentials and the number of packets released. This is a little more complicated than the method that Dave uses, which is CMAP immunity, but it's complementary. And at the time, we didn't know that Dave was around, although he was in the clinic, I think, but we hadn't run into him. So we used this with Mark Rich to show that the neuromuscular junction had completely recovered when you gave gene therapy. And I would emphasize this point. When you put a paper in, survival is all very good. But you want to know that you've corrected the motor neuron. This shows you've corrected the motor neuron, because the neuromuscular junction is completely normal. Again, I just want to make the point that in every case, the SMN levels are actually below the carrier state. So the SMN level is not massively overexpressed in these cases. We also did intrathecal delivery, and this says intrathecal delivery, but it's not actually intrathecal, it's ICV delivery into the mice. This actually increased the mice actual survival. I'm just going to draw your attention to this. The median at the top dose, which is 3.3 times 10 to the 10, is 282 days. If you go to either the paper that reported toxicity of SMN, which is from Livio Pellizzoni's group, or the original paper, they find that 5 times 10 to the 10 vector genomes, it's affected, if we do vector genomes per gram, it's 5 times 10 to the 10 per gram. It's more if you do it in kilograms. Mice usually do not weigh in kilograms, so I've got kilograms on this just for the convenience. But these mice only live for 156 days. So I just want to make the point that you can deliver into the CSF and get very high survival. We find actually when we reproduce this that we go over 300 days with the median survival. And also, if we take the SCAAV CBA, the actual clinical construct and promoter, and do exactly the same things that they've done with the GUS-B promoter, and we look at the M wave and H wave reflex, there is no abnormality. There's no abnormality in clasping, no abnormality in behavior, no abnormality on the rotor rod, and no abnormality in electrophysiology. And that's at 300 days after injection. So this toxicity does not exist with the clinical vector. It may exist with the GUS vector, but nobody's using it in the clinic, so what's the point of knowing that? We also developed a pig model. And we often get asked, and I've had the question put to me, why do you need to do the pig? It got approved by FDA with just the mice in the application. Yes, but somebody's got to fund the clinical trial. And the way the money was developed by Avexis and got through the investors was due to the pig model, because they saw the pigs and the results I'm going to show you, and that resulted in the investment. So in this particular vector, we have the CBA promoter driving GFP, and we have an H1 promoter driving the construct that knocks down the pig SMN. And also in review, we got a question that were we worried about the toxicity of knocking down pig SMN? I restrained myself from replying that I believe I don't have any pig genomic DNA in me that I'm aware of. I might eat some pig DNA, but I don't have any in me. So that question was relatively easy to answer in the first review. And so we did that. And then we rescued with AAV expressing SMN. And the knockdown occurred at 5 days, and then the pigs developed weakness and a splay gait at 24 to 34 days. Then they get worse, and they get worse, and then the vets at this point at 46 to 69 days come after us and say, you have to sacrifice this. And you're also at this point going in at night and feeding the pigs, because otherwise they get kind of very excited and they like their food. And this is delivered via the cisterna magna, as indicated here. And this is actually what the pigs look like. So the control goes first, hopefully, yes. There is a reward at the top of the ramp, so marshmallows or other goodies. They are very fond of marshmallows, pigs. And if you have no rescue with just the knockdown, you can see the pig has some difficulties getting up. In fact, it doesn't get up the ramp. We do give it the marshmallows, so it's not deprived. And if you actually then look at the early rescue, so early rescue would be the day after the knockdown, so very early. And the pig runs up the ramp fine and gets the delicious marshmallow. And then if you do it at the first sign of symptoms, which is what we call late rescue, then the pig has got much better movement and much improved, but it's definitely different than the early rescue. And this is also reflected in electrophysiology. This shows the CMAP, but I want to draw your attention to the MUNI. Some letter, I put these letters below, but anyhow. And you can see with the early rescue in orange that the MUNI is completely rescued, but that's not the case when you do late rescue. In other words, once the motor neuron's gone, it's gone. You can't rescue it by giving it SMN. That doesn't help. So the thing I want to emphasize is timing is critical. So once you get in here and you've got all your motor neurons, you can fix it pretty good. If you go later, it's less good, and late is pretty mediocre. That's from the animal models. And the same is true in patients. If we go to the clinical trial of a VSMN, this was done at Nationwide Children's by Jerry Mendel. So you don't want me going anywhere near a patient. Keep me well away. And so this simply shows the two cohorts. One's a lower dose, what is a higher dose, and prednisone was administered to suppress the immune response to the virus, but this was only done after the first patient, because the first patient had a clear response with the liver enzymes going up, et cetera, and so that's why it got introduced, and the FDA allowed it. This trial had to be done on patients that were evaluated by the physician to have symptoms. So they're not meant to be pre-symptomatic. So it could be up to 6 months. Obviously had to have 2 copies of SMN2 and a mutation in the SMN1 gene, and there were various exclusion criteria. So the priority was safety and tolerability, and then the time from birth until death, etc., was secondary. And then the CHOP-INTEND score, which is simply a behavioral motor score for how well these patients are doing. So if we look at the natural history of SMN, because this is controlled by the natural history trials, this shows the normal milestones of a child in the bottom. And then if you actually put what happens with SMA, so first of all, 90% of SMA type 1 patients will not survive to the age of 2. So 8% survival at 20 months, 25 at 13.6, and 50% at 10.5 months. And so you can see there's a very characteristic survival. There's also the ability to move the head. There's the ability to roll over. There's these criteria that you can be used for assessment. This shows the CHOP-INTEND in the trial. And the outcome or death or surrogate is if you get 16 hours ventilation dependence, then that's counted as death. So you can see that these patients are past the 8%. They're surviving much longer than would be expected in all cases. And they have much better function in their, in their actual CHOP-INTEND score. So you can see these are the 2 earliest treated patients, which hit the plateau, so they're actually doing very well. And these are the other patients, and this is the natural history course of the disease. Later treatment, you can see here, doesn't do as much good. And 4 were able to stand, and 2 were able to walk. And this actually shows the phase 3, which I'm not going to go through in detail, but it's basically equivalent response. So the phase 3 duplicated in a multi-center trial the response from the phase 1. And then this is the youngest kid that was treated in the trial at 1 month. And obviously, now he's now on his mission. He's going to his office. Here he goes. Oh, the elevator button, the actual prize of the situation. So he's got the elevator, yes, very happy. The other thing I will say about this particular child is, how do you know your clinical trial works? Well, just bring in Dave Arnold to do the electrophysiology, and the child will immediately run away at top speed down the corridor of the hospital. In the phase 3 trial, 2 patients actually died, not from the gene therapy itself, but from other causes related to the fact that this is a symptomatic trial in SMA. And these are the 2 patients, so patient 1 and patient 2. So you can now take all these different tissues and you can look to see if they have vector genomes on them. This simply shows the vector genome copy number for all these different tissues in the 2 patients. So the AAV 9 is getting many places, not just the liver, but it does definitely hits the liver. And if you actually look at what they named it preclinically, which is a nosmogene apophorovac, which is a horrible name, you can see if you look at digital droplet PCR to quantify the copy numbers that it's got 2 copies in the motor neuron. If you do immunohistochemistry on the treated patients, and this is from the cervical cord, you can actually see the motor neurons in the brown that have been taken up the AV and produce SMN, so this is an SMN stain. The non-treated SMA subject, which came from our historical set, basically has no motor neurons and basically also doesn't stain for SMN. This is a subject which is a non-SMA subject that was in our collection that is an age-match control. You can see that this has clearly positive motor neurons. Then these are darker than the ones in the patient. If you actually quantify the amount of expression, which is shown here, if you use the unaffected control that has 2 copies of SMN1 and 2 copies of SMN2 and set that at 100%, then the patient has approximately a 25% recovery of SMN above the 2 copies of SMN2 because you've normalized the 2 copies of SMN2 out of it. So that's 25% increase plus whatever amount of SMN is produced by 2 copies of SMN2, so approximately 35% of the SMN. So again, you can see that this is not overexpressed. This shows the relative expression in other tissues, as shown here, relative to GABDH. Okay, can we do anything else to further improve gene therapy? Well, one thing is we could actually modify the SMN2 gene, either by targeting regulatory regions, or even more likely is to actually target the base pair change, and to change this T that's in the SMN. And for this, we collaborated with David Liu's lab and Amanda Arab. They did all the base editing stuff and developing of the base editors. And so we tried it. How well does this work? Okay, so you edit the C to T, and yes, you have to have the vectors, because of the size of the Cas9 that you need to bring the guide RNAs in, you have to split the vector into 2. So the base editor is split between 2 vectors, and then one of those vectors also has the RNAs that are produced in it. They have an intin, which is going to allow the protein to reform in vivo. So this is what this is, and this is relatively efficient when you're looking in cells in culture and doing the transfections. If you actually look at the frontal cortex, you can also show that it's relatively efficient in neurons. Do we have the numbers for a motor neuron? The answer is no. If you look at the electrophysiology, I'll come to that in the next page, you do actually get correction. And that is shown here. You can see this is uninjected SMA animals for their muni. This is the ABE corrected, and this is the heterozygote. You can see there is being correction and rescue of the multi-unit number. The survival, however, is really, it's improved, it's extended, 19 days, it's about 4 days. It's not very impressive. And this just shows that the muni is improved as indicated here, which I've already said, but the CMAP is not vastly improved. The survival, so what we did was give low dose of risdiplam with this to try and extend the survival, with the idea that the low dose of risdiplam would compensate for it. However, when you do that, this is the green line. They get worse, they don't get better. So adding risdiplam to the ABE editing makes it worse, not better. Again, that's also true of the CMAP and Muni. It doesn't help at all. If anything, it decreases it. So there's a toxicity when you put the two together, even at a low dose. And if you do it at a high dose, the animals that live the longest are the ones that just have risdiplam. So vascular delivery of a nosmogene aberrant or gene therapy is basically effective in SMA. The expression of mRNA is abundant in liver and muscle, but it's found expressed in brain and also in the spinal cord. There's no there's about 1 to 2 copies in the CNS, with the motor neurons having about a 25% increase. And we've detected no toxicity with old nosmogene aberrant, and it does result in increased SMN in organs, and the 5.6-year follow-up is actually quite good. So go to the next one. So this will be very quick. So we decided one thing that might be necessarily is to get treatment for symptomatic patients. So why not try treating symptomatic mice? So we took mice at different time points, 2, 4, and 6, and gave them combined treatment or single treatment. When we do that, we find the SMN is increased when we use the combined treatment in all cases, and that even at P6. And when we look at the survival, which is actually just draw your attention to this here, is that at P6, when you add the ASO and the drug that induces the thing, you get more SMN and you get better survival. And this is approximately equivalent to the single treatments at P2. So double treatment does seem to be more effective. This is still relatively early in the disease. And so we would conclude that dual treatment is an advantage, and that maintains the electrophysiology. Now I'd like to just finish with the what SMN does and where the mechanism and where we're going with it. So SMN is known to assemble the SM ring onto snRNA, and that's shown in this slide. The snRNA then gets imported into the nucleus and does splicing. The level of SMN assembly in rescued animals or milder animals does correlate. So the more severe the animal, the less the snRNP assembly, the better and increased assembly, the milder the animal. So fine, we know that this is one function of SMN, but how can we look at whether this is the critical function? Because SMN has been reported to have multiple functions. So what we've done is taken a cell line that lacks SMN on one allele, has SMN knocked out on another allele, not knocked out, has it floxed. So when we add Cre, this removes SMN, and this results in death of the cells. So we successfully killed the cells. That doesn't sound very exciting. So this shows that that is the case. You take this, you seed it out, and actually if you put in all these different missense mutations, none of them rescue. Not one of the missense mutations rescues. But what we can do is go a step further. So we want to discern all these multitude of functions that SMN is supposed to do. And we want to ask whether those are actually important. So the first one is loss of exon 2B. The 2B exon contains the binding site for alpha COP. And so this simply shows an NSC34 cell line that we didn't make, but we characterized, that lacks exon 2B. We then made cell lines where we tested the SMN that lacks exon 2B. We show this SMN lacking exon 2B is viable and does result in cells that live. They also perform SNRMP assembly. So that means that this is a viable SMN in terms of SNRMP assembly, but is missing the alpha COP binding site. What I will say on this, and I'll give it away, is that the mice, we've made mice now that have that gene knocked out and it's completely viable. And if we take it and we do another experiment with these cell lines where we do exactly the same thing, but we introduce SMNE134K, this is not viable, remember, and we mutagenize it, we can isolate suppressors. One of these suppressors is in one of the SM proteins, D37N. We also have another one that seems to be completely independent of SMNE134K. So this simply shows the suppressors here, the D37N, and this shows the fact that 2B mice, or actually not mice, but cells lacking in the 2B exon don't bind alpha COP. The mice are completely normal. We can't find anything wrong with them. They run around, and we can put it against the deletion allele. It doesn't have to be 2B minus minus. It can be 2B minus across from a knockout. We are breeding the mice now to determine whether SMNE134K can be suppressed with SMND37N. It's been a complicated experiment, more difficult than we expect, but this can resolve the actual suppression, and it is due to the SNRMP assembly. So I'd simply say that genetics can be used to dissect which function is critical. And this is summarize it, which I'm going to skip. And then I want to acknowledge all the people that have worked on this. So these are the various knights through the age. You can see Dave here. I'm not going to list everybody and what they all did. And obviously the clinical team was quite large. Brian Casper is a very good colleague of mine, and it was a great collaboration with him. So thank you very much. Thanks.
So we have just really not much time.
Yeah, no,
according to the way Arthur always does, he doesn't always follow the rules, and I think that's the reason why he's been able to really make such a big impact on the field. I mean, really, he doesn't have a big ego, so he really doesn't highlight how he really moved this field forward through collaboration, offering the resources of his mice, which are used all across the world and actually have fed into all three therapies. So he really undersells it. So, but any quick questions? Do we have questions online? Yep.
Yeah, so is there a consensus on the evolutionary reason for why humans have the SMN2 gene?
There's no consensus on it. What I would say is it's a relatively recent event, and there's no selection for retaining SMN2. And if you look at the actual mutation rate of SMN2 in the Caucasian or European population, it's very high that SMN2 gets mutated and deleted. So I would say that it doesn't have much chance of surviving if we live another 1,000,000 years or however long it takes to get that far. If we evolve, we'll see. I mean, a bit sarcastic, but
maybe one more question.
How old are the oldest human patients who have received the treatment in the clinical trial?
Depending on which clinical trial you're talking about, I think the patients from the nusinersen must be around 10. The gene therapy patients are around 8 from the phase 1 trials.
Great. I think due to time, we're going to have to hold on questions, but Arthur will be here afterwards if you have some questions, come by. Before we wrap up, Arthur, I wanted to share a token of our appreciation for sharing your work and the contribution that you've made to the field of neuromuscular
What's
medicine.
the state? I'm trying to work this out.
It's where you're standing right now, the state of Missouri. And so shortly after I arrived to the University of Missouri, we treated the first patient with gene therapy for SMA. So I saw those infants every 3 months through this trial, and to be able to see it impact people in the state and really across the world is amazing, and it's because of collaborative science. Dr. Lorson really has contributed massive amounts as well, and so I thank you both for your work.
Thank you.
So please join us next month. We're actually going to be in Kansas City for Dr. Friedman, and so there'll be an in-person viewing here as well as in Kansas City. Thank you.